I'm delighted to introduce Jessica Salvatore, who is Associate Professor of Psychiatry at Rutgers. She received her PhD in child psychology at the University of Minnesota, and then was a postdoc at the Institute for Psychiatric and Behavioral Genetics at Virginia Commonwealth University, where she began her faculty career as well before moving on to Rutgers, 3 years ago or so. And I will leave it at that because I'm going to let her tell us more about what she's interested in, but I'm very excited to listen to her talk today.
Wonderful. Thank you so much, Rachel, and for all of you for welcoming me here. And I'll be talking today about a series of studies looking at indirect genetic effects and the development of alcohol and drug use disorders. And just want to be clear that I don't have any conflicts of interest to declare. And of course, I'm the one speaking today, but I want to be sure to acknowledge and show my appreciation for the team that really makes this work possible. So at Rutgers, I lead the Genes, Environment, and Neurodevelopment in Addictions program, which includes Dr. Sally Kuo, an assistant professor, Megan Cook, a research associate, and Erin Lumpy, who is a graduate student in our group. And our goal is to understand how alcohol and other substance use disorders shape and are shaped by close relationships across the lifespan. And in addition to our sort of nuclear group, we work in a highly collaborative environment, as many of us do in the area of psychiatric genetics. And the two groups that I especially want to acknowledge for the work that I'm presenting on today are the collaborators through the COGA project, the Collaborative Study on the Genetics of Alcoholism, especially Danielle Dick, who is my postdoctoral mentor, Fazil Aliyev, Nate Thomas, Sarah Brislin, Peter Barr, and Jackie Myers, probably some familiar names to those in the group here. And then the second sort of group of collaborators I want to acknowledge and thank up front are Dr. Ken Kendler and the Swedish National Registry collaborators, including Jan and Christina Sundquist, Sara Larsson-Lahn, and Henrik Olsen, as well as Alexis Edwards. And I would be remiss if I didn't thank the support that I and my group have had over the years from NIH as well as the Templeton Foundation. So the work that I'm presenting today is really a team effort and reflects that team. So as the backdrop for my talk today, I wanted to start with a story about the transformative power of close relationships as told in the children's book, The Pout Pout Fish. So for those of you who may not be familiar with the Pout Pout Fish, this is a story of Mr. Fish, who, despite the efforts of his ocean-dwelling friends to cheer him up, maintains that because he's a Pout Pout fish with a Pout Pout face, his destiny is to spread the dreary juries all over the place. But this all changes one day when Miss Silverfish comes and plants a kiss upon his pout, and then she swims away. At which point, Mr. Fish realizes that he's in fact a kiss-kiss fish with a kiss-kiss face for spreading cheeries all over the place. And I like to start with a story like this because it really sets the stage for the program of research that I and my collaborators engage in, which is trying to understand how relationships impact risk for substance use disorders. And across the three data stories that I'll be sharing with the group today, I'll be making the case for indirect genetic effects, and I'll sort of ground that concept in the next couple of slides here. But just to give you a preview of where we're going to be tracing indirect effects across the lifespan, starting first with nuclear families, so within parent-child relationships or nuclear family trios, how family adversities transmit genetic risk across generations. Moving into adolescence to look at peer influences and how adolescent peers' genetic makeup influences risk for substance use disorders. And then we expanded in this particular project to also consider major depression and anxiety disorder. And then finally, we will be in the sort of the third act of my talk today. I'll be talking about partner influence and how spouses' genetic makeup influences risk for drug and alcohol use disorders. And feel free to interrupt me at any time. I've also left plenty of time at the end for questions. So I'm also very happy to answer questions along the way. All right, so let's talk first about what we mean when we're saying that we're looking at indirect genetic effects. And we're going to start that story by first talking about what we mean when we talk about direct genetic effects. And this is really where most of the sort of genomic partitioning space is sort of centered upon. So what I've depicted here is the standard quantitative genetic partitioning model, or a direct genetic effects model for a phenotype. And I've just, put here an alcohol use disorder phenotype right here. And typically what we're doing at a population level is we are partitioning the variation in our phenotype, there's my cursor, into an additive genetic component and then an environmental component. Now, when we're talking about indirect genetic effects, and they're sometimes called social genetic effects, what we're doing is we're expanding from a sort of single person centered approach to thinking dyadically or even beyond to consider social networks. And that's really the where we'll be headed in the second part of my talk. So here I've expanded the model to consider two people, person one and person two. They each have their own phenotype that can be each sort of independently partitioned into additive genetic and environmental components. So for person one, and then also for person two. But of course, for any of us who've been in a relationship, we also know that the phenotype of person one becomes an environment for person two. And so that is denoted with this, this sort of unique environmental component that comes from person one all the way down to become an environment for person two that in turn is impacting the phenotype of person two. And if we follow our path tracing rules from structural equation modeling, what we can see is that if you trace back from the phenotype of person two, back through the environment that person one is providing them, up through person one's phenotype, and all the way back to person one's genotype, that is the indirect genetic effect pathway that we are tracing that I'll be talking about throughout the data stories that I'll be sharing with you today. All right, so turning now to our first empirical example, looking at a genetic nurture effects for alcohol use disorder. What we're doing here is looking at whether and how family adversities transmit genetic risk across generations. And this work was led by a graduate student in my group who's since graduated on to postdoc, Nate Thomas. And so just want to acknowledge him here before we head into sort of the content. So the framing question for this work is, how is genetic risk passed from parents to children? Now, the conventional understanding, sort of consistent with that direct genetic effects model that I presented on an earlier slide, is that parents transmit risk alleles to their children across generations. But we also know that allele sharing represents only one potential mode of transmission of genetic risk within families. And that's because the parental genotype also influences the family environment that children experience or are exposed to. Now, the family environments that we focused on in this particular study were looking at the experience or exposure to parental divorce and parental relationship discord. Now, the reason that we focused on these two particular forms of family adversity are both that they're relatively common. They can occur independent of one another. Not all highly discordant relationships end in divorce, and not all divorces are of high discord relationships. And importantly, our group and other groups have demonstrated that genes that contribute to the experience of divorce and relationship discord also overlap to a fair degree with the genes that influence alcohol and other substance use disorders, making these a really good candidate environment for understanding these family adversities as sort of a environmental mediator of genetic risk across generations. So to look at this question, we used the collaborative study on the genetics of alcoholism sample, COGA. And I know many in the room are familiar with the COGA sample, but for those who are maybe less familiar, this is a clinically ascertained family-based sample of originally treatment-seeking probands were recruited from outpatient treatment facilities, and then their families were also invited to participate if the families were sufficiently large with 2 or more members in the COGA catchment areas. And there were also a subset of comparison families who were recruited from the same community, so not ascertained based on alcohol use disorder status, but as we know, out in the community, there are families affected by alcohol use disorder. So the comparison families were not necessarily unrepresented in terms of alcohol use disorder, they just weren't selected based on their AUD phenotype. Now, within this larger COGA family-based sample, we selected parents and children who we confirmed via genotyping with relevant phenotypic and genotypic information available to sort of facilitate the analyses that I'll be talking about. And we focused, or what I'm focusing on here in the presentation today is on the results from the European ancestry subsample. So this was just under 5,000 individuals of European ancestry. Roughly 50% of the individuals in the sample were female, and their average age at assessment was just under 31 years of age. So, in order to examine the nature of nurturing COGA, we examined a series of alcohol use behaviors in the child generation, ranging from the most sort of clinically severe presentations of DSM-5 alcohol use disorder criteria to a still severe but sort of less clinically sort of significant indicator of lifetime maximum drinks in 24 hours, which are also genetically correlated with alcohol use disorder criteria and disorder. We also looked at age at alcohol initiation, as well as age at first alcohol intoxication. So we're really spanning sort of the range of alcohol-related milestones across individuals' sort of drinking careers, ranging from initiation all the way to development of problems. Within the parent generation, we looked at their genome-wide polygenic scores for alcohol problems, and I'll talk a little bit about how we generated those scores in a second here. And then we have a measure of parental relationship discord, whether or not the parents experienced divorce or permanent non-marital separation, because not all of the families were legally married, or not all the parents were legally married in the sample. And then we also covaried or statistically controlled for the corresponding alcohol use behavior in the parent generation, as we are looking at in the child generation analyses. So when we're looking at child DSM-5 alcohol use disorder criterion counts, we're also covariating for the parents' AUD criterion counts. So controlling for the parents' phenotype in addition to these sort of family adversities that were of key interest. Now, our genome-wide polygenic scores were derived from summary statistics from a large genome-wide meta-analysis of DSM-4 alcohol dependence from the PGC, GWAS, Audit P from the UK Biobank, and then from MVP, the AUD ICD codes. And then our indirect effects models were fit using structural equations modeling, and I'll walk us through what those models were on the next slide. So in this next sort of set of pictures, or the graphic that I'll show, it's really building this indirect effects model, looking at how studying families can help illuminate the nature of nurture and how parental genotypes also inform the type of environments that they're providing to their offspring, which in turn may inform or contribute to the offspring's alcohol use behaviors. So our outcome of interest is the offspring alcohol use behaviors, again, ranging from our age at initiation measure all the way up to our clinical criterion counts for DSM-5 alcohol use disorder. And up at the sort of most distal level, we have our parental genome-wide polygenic scores. So we have separate genome-wide polygenic scores for moms and for dads. And these are contributing directly to the offspring's own genetic risk for developing alcohol problems. But our key question was really trying to understand whether and how the parent genetic predispositions were also contributing to the likelihood that the family would experience marital disruption or marital discord, and whether that in turn was associated with the offspring alcohol use behaviors. So really, again, tracing this pathway from the parental genotypes through the family environment and down through the offspring alcohol outcomes. And so what we found, consistent with our hypothesis, is that parents transmit genetic risk for alcohol problems to their offspring, both directly, as denoted by those black lines there, but also indirectly via genetically influenced aspects of the home environment. And that's what's denoted in the blue paths here, from the parental genotypes through the likelihood that the family would experience disruption or relationship discord, which in turn was associated with earlier age at first drink, earlier age at first intoxication, higher max drinks in the offspring, as well as a greater number of DSM-5 alcohol use disorder criterion counts. And so our evidence for indirect genetic effects through a parental relationship discord was observed across all of these alcohol use behaviors, ranging from initiation to clinically significant symptoms. And there were much more, I would say, modest effects for our parental relationship or our parental divorce or non-marital separation mediator. These were only really associated with age at first intoxication as well as age at first drink. So some time-specific effects, it seemed. Mm-hmm.
So did you measure parental discord as a continuous measure?
Yes, well, it was a sum score of 4 different items that went into the measure. It was a weighted, yeah, I mean, it didn't range from 0 to 4 because they would weight things because the scales were different. But
more informative than the binary divorce.
Yes.
Which may explain better.
Yes, absolutely. And I think too what this might be tapping is just that divorce, divorce is definitely a process. It's not a discrete event, even though there's sort of a date at which it happens or like people report at which it happens. But I'm guessing that divorce might just have more time limited effects in terms of disruption to like parental monitoring or sort of, restricting children's or offspring's access to alcohol. Whereas discord does seem to have a more pernicious and sort of long-term effect on the offspring's alcohol outcomes. But yeah, measurement or psychological, I think, both. Mm-hmm. Okay, so in that first example, we were talking about the indirect genetic effects as transmitted within nuclear families. And now we're going to transition a bit to talking about adolescent peers, looking at how adolescent peers' genetic makeup influences risk for the development of substance use disorders, major depression, and anxiety disorder in adulthood. And this is a collaborative project with the Swedish National Registry collaborators. And I want to just make a special acknowledgement of Henrik Olsen, who is the biostatistician on this project. So let me share a little bit about the Swedish National Registry data. So these are population information maintained by Statistics Sweden, and it's facilitated via record linkage via the unique 10-digit ID that's given to all individuals in Sweden, either at birth or immigration. So something akin to our Social Security number. And from these population level databases, we're able to ascertain and learn a lot about the individuals in the Swedish population. So using multi-generational pedigree databases, we're able to link parents to their children, and we're also able to link spouses to one another. We're also able to ascertain individuals' number of years of education, their income, their residence, as well as the schools that they attended while growing up. And then in terms of our psychiatric and substance use disorder outcomes, we're able to look at their registrations within the national health registries across a series of medical, legal, and pharmacy registers. So in contrast to the data example that I just showed you from COGA, where we used a genome-wide polygenic score to index individuals and families' genetic risk for alcohol use disorder, in the Swedish population, we have the benefit of a massive sample size because we're looking at the entire population. But of course, at that scale, there's not genome-wide information. And so we have to find alternative ways to index genetic risk to sort of facilitate these types of analyses. And so something that Henrik and Ken Kendler have developed over the recent years, and that's implemented as part of this project, are the generation of pedigree-based family genetic risk scores. So like a genome-wide polygenic score, these FGRS, the family genetic risk score, which we for short call FGRS, are personalized indices of genetic risk that are inferred from diagnoses in first through fifth degree relatives and weighted by degree of genetic sharing. Oh, sorry. Was something saying? No? Okay, sorry. I thought there was a question. And so again, these are pedigree-based estimates of how much genetic loading one has for a disorder. And so a high FGRS represents an individual either from a densely affected pedigree, and it can also reflect an individual with like a high number of affected first degree relatives. So just in this toy example here of a pedigree here, where red indicates an affected individual and blue indicates an unaffected individual, someone like this person would have a higher FGRS score because they have several affected first-degree relatives, 1, 2, 3, in contrast to someone like this, who does not have any first-degree affected relatives. And so this just kind of gives a visualization, I hope, of how these sort of weightings are happening across extended pedigrees. And just to also provide some additional phenotypic context on why we would even take a look at sort of a genomic perspective on social network influences on health. So we know from decades of research in the developmental psychology literature, and I'm a developmentalist by training, that adolescent peers influence a range of outcomes, including behavior problems and depression. So on a phenotypic level, we know that peers influence one another. And more recently, we know from research conducted in the AdHealth study that classmates and friends' genome-wide polygenic predispositions to smoke predicted adolescent smoking behavior above and beyond adolescents' own polygenic predisposition. So this was a sort of genome-wide polygenic scoring approach, providing some initial evidence for these indirect social genetic effects, looking at peer influence and peer indirect genetic effects on, in this case, smoking behavior. And so we were building on that, using our tool of the family genetic risk score. And our question here was whether peer genetic predispositions influence likelihood of registration for a drug use disorder, which I use the acronym DUD throughout the rest of this section, alcohol use disorder, AUD, as many of you know, major depression, MD, and then anxiety disorder, AD. And so within the Swedish registries, based on the information that we're able to sort of map to individuals, we were able to make 6 peer group classifications, 3 based on geography and 3 based on school-based affiliation. So if we think about any one individual, we're able to map that person to the 1,000 meter square, which is roughly 10 blocks area that they grew up in, which is further nested in what's called a demographic and statistical area, a DESO. And a DESO really refers to geographically bounded areas. So things like, oh, you're in an area that's bounded by like this busy road and this river and, these train tracks kind of thing. And then municipality is the largest geographic unit that we were looking at. And this is something akin to a county-based level, here in the US. We also know which lower secondary school individuals attended between ages 7 and 16. And then we also know information about individuals' upper secondary school. So within the Swedish system, upper secondary school is the non-compulsory part of education, and people can either track into a college preparatory track or a vocational track. And people attend these schools, or the upper secondary schools, between roughly ages 6 to 18. Some individuals continue until they're 19, and that has to do with the length of that upper secondary school program. At some point historically, some of them were 3 years. They've all, I think, transitioned now to 2 years. And then these vocational and college preparatory programs are housed within a sort of single upper secondary school unit. So we know which program people are in, and which, who their peers are within those programs. But we also know like which generally building they're in with their upper secondary school peers. And just to give an idea of how many peers are actually contributing to these social or indirect genetic effect scores at the municipality level, which are our largest geographical level. We have 121 peers on average who are contributing to the peer genetic effects. And then our smallest unit are that 10 block radius unit. There's on average 2.5 peers who are contributing to these genetic risk scores for the peer genetic effects at the geographic level. And then at the school-based level, the type of program, within the type of upper secondary school program, it ranges from, about 10 students contributing to these social genetic effects. And then at that lower secondary school level, roughly 25 individuals. So just to kind of give an idea of the number of individuals contributing at each of these levels. Yep.
Is this time-based? So are these like the various groups, people that are in that school at that same time kind of thing?
Yes, so it's tracked to birth year.
Okay.
And so yes, like that's how we know, or that's our best guess. I mean, some people based on their birth year are going to be, you know, redshirted or whatever into kindergarten, or I mean, this isn't kindergarten, but you know, there's probably some people on the edges who might be incorrect, but by and large, they're going to be.
Okay. So when you're getting down to these groups and so on, this is kind of people that were in these same places at the same times.
Yes.
Okay.
Yep, that's right. So our outcomes were proband registrations between ages 17 and 30. So we have our drug use disorder registrations, alcohol, anxiety, and then major depression. And I've noted in italics here the rough the percentage of people who are becoming registered for each of these disorders within the these medical, legal, and pharmacy registries. And so you'll see that we have, you know, the smallest N is under our alcohol use disorder registration, and then our largest N is for anxiety disorder, where roughly 15% of people became registered in this period between ages 17 and 30. And our key predictor were these peer FGRS scores for each of the 6 geographic and school-based classifications that I mentioned on that previous slide. So we have a peer FGRS for drug use disorders, alcohol use disorder, anxiety disorder, and major depression. And so when we look at this, and I'm sorry that this is trying to make this minimally in our way, but I'm not sure how to minimize it. So when we look at the association between peer FGRS scores and proband's risk of registration, we find that indeed the peers, you know, polygenic predispositions or family genetic risk scores for each of these disorders is associated with the likelihood that the proband goes on to be registered for one of these disorders in adulthood between ages 17 and in young adulthood, ages 17 to 30. The analytic sample size for this was just over 655,000. And I'm going to walk us through the sort of set of results in this next graphic here. So, so this is sort of a visual depiction of the take-home point that peer FGRS are associated with proband risk for registration. So along the x-axis here, we have each of these 6 school and peer school-based and geographically based peer group classifications ranging from municipality to this 10-block square radius, lower secondary school, up to the type of, within the type of upper secondary school, that program, vocational or college preparatory. And then within each of these blocks, we're indexing the association between the peer FGRS for each of these disorders, in blue denoting drug use disorder, in orange alcohol use disorder, in gray anxiety disorder, and then in yellow major depression. And so each of these bars here is indexing that association between the peer genetic, family genetic risk score, and then the proband's risk of registration for that same disorder between ages 17 and 30. So there's just a couple of things that I want to highlight for the group here. The first is that within our geographic-based definitions of peers, we find an association, but it's pretty modest. You see it sort of bouncing around roughly somewhere between an odds ratio or a hazard ratio of just over 1 to about 1.05. And there's really not a lot of differentiation between in the predictive power across the different disorders, as you'll see by the large and rather overlapping confidence intervals for these estimates. And where we start to see more of the action and the predictive value is once we get into the schools, so our school-based peers. The lower secondary school, I would say the effects are quite in line in terms of the effect size with what we saw for the geographically defined peers. But really, once we start getting into later adolescence and the upper secondary school, and then especially within the program that you're in within upper secondary school, that's when your peers really, your peers' genetic predispositions really start to matter. And we tended to see the strongest effects for the peer genetic predispositions for externalized disorders, so drug and alcohol use disorders. And I just want to say, too, that we tried to make these effects go away through statistical control for a number of covariates. And these effects sort of were robust to these many statistical controls, including the proband's own family genetic risk score for these same disorders. Sex, because we of course know that there's sex differences in the presentation and frequency of these disorders. Whether or not the proband had a registration for any of these disorders prior to age 16. Individual socioeconomic status indexed both through family income, family educational attainment, whether the individual's FDRS score for educational attainment, because we know that there's quite a bit of genetic overlap between educational attainment or propensity for higher educational attainment and lower likelihood of experiencing one of these, especially the externalizing drug and alcohol use disorders. We also covariated for which type of upper secondary school program they attended, whether it was vocational or college preparatory. And then importantly, we also controlled for the peers' actual phenotype. So above and beyond the peers' phenotype, we still continue to see an indirect effect or an effect for the indirect or social genetic effect for the peers' genotype. All right, so we've talked about parents and nuclear families. We've talked about peer transmission and how peer genetic predispositions influence risk for these disorders. And now I'd like to transition to talking about partners or spouses. So we're looking here at how a spouse's genetic makeup influences risk for drug and alcohol use disorders. And the first example that I'll share is again from the Swedish National Registries, and we were focusing on the development of drug use disorder. And just special recognition again for Sarah Larson-Lahn, who is the biostatistician for this project. So our question here was, is spousal genetic makeup associated with the risk of developing drug use disorder during marriage? So what we were using again were these the family genetic risk scores for drug use disorder. And our question was above and beyond the direct genetic effects, so the association between my own genetic risk and my own likelihood of developing a drug use disorder during marriage, does my genetic predisposition influence the likelihood that my spouse develops drug use disorder during marriage? And so this pathway here is indexing the spousal indirect genetic effect. And just to sort of give some additional context, our drug use disorder phenotype here was inclusive of opioids, sedatives, cocaine, cannabis, amphetamine, and other psychostimulants, hallucinogens, and then other drugs of abuse not otherwise specified. Within the registries, we were able to identify just under 300,000 couples. And I also just want to share that, even within this large sample size, probably for everyone's good health, there are not many people who during the course of their marriage are becoming registered. So I just want to be transparent about that. So just under 1,500 men became registered for drug use disorder over the course of marriage, and just under 1,000 women were registered for drug use disorder during marriage. And what we find is that spousal genetic makeup is associated with the development of drug use disorder during marriage. So I'm going to walk us through these results here. So along the x-axis here, we have our proband grouped by female probands and then male probands. And then within each of those groups, we have two different types of the family genetic risk scores that we're indexing. We have the direct genetic effects, so the association between my own genetic risk and my own drug use disorder phenotype during marriage. And then in the purple, we have the spousal FGRS, so indexing the association between the spouse's genetic predisposition and the likelihood that their partner develops drug use disorder during marriage. And so what we find is an association between the spousal FGRS for drug use disorder and proband registration during marriage. And as we would expect, this indirect genetic effect it's statistically significant, but of course it's more modest than what we see in terms of a direct genetic effect. So we would, under no model would we necessarily expect that the indirect genetic effect would be stronger than the direct genetic effect. And importantly, in this type of dyadic modeling, we're accounting both for the correlation between spouses' genetic risk scores, because we know there's both phenotypic, but also some degree of genetic assortative mating within spousal dyads. And the model is also accounting for the phenotypic association between partners. So that, spouses resemble one another in terms of almost every single phenotype. And similar to our peer genetic effects analyses among the adolescents, we also controlled for the spousal drug use disorder phenotype. And when we do that, we continue to see evidence for the spousal indirect genetic effect. So there's something unique that's being picked up with this spousal family genetic risk score that's not, overlapping or sort of identical to what's already being picked up by the spousal phenotype. So there's something distinct there. It's not just a pure mediation through that indexed phenotype. And then this last data example that I wanted to share with you is an expansion of this work looking at indirect genetic effects within spousal dyads, focusing in this case on alcohol use disorder and taking a polygenic approach. And this is work led by Dr. Sally Kuo in my group. And we are again using the COGA sample for this last study that I'm sharing with you. And so just to say a little bit more about the marital dyad sample in COGA, it includes 666 opposite sex marital dyads, so 1,320 individuals. And again, for right now, I'm just going to focus on the individuals of European ancestry because we are using those genome-wide polygenic scores. And these were individuals who were in their first marriages based on individuals' sort of concurrence regarding their age or reporting the same year at first marriage. Within the COGA sample, there's actually quite a variety of marital histories that participants have. And so just for simplicity, we're focusing on first marriages because higher order marriages have certain complexities that we need to start somewhere with these analyses. And so for first marriages, it was. The average age at assessment for these analyses was roughly 50 years of age for both the males and the females in the spousal cohort. And then just to give sort of a flavor for the educational attainment and sort of other sociodemographic characteristics in this marital dyad sample, most individuals had completed high school and had a couple of years of college. So similar to what we saw using those latent or inferred family genetic risk scores in the Swedish national registries, we found that when we used a polygenic approach indexing polygenic risk for alcohol problems, that marriage to a spouse with a higher polygenic score for alcohol problems was associated with a higher alcohol use disorder criterion count during marriage. So that again is indexed during this spousal or indirect genetic effect path, which is, roughly a third to a quarter of what we see for the direct genetic effect path. So sort of falling in line is sort of similar to what we saw with the Swedish registries, evidence for effect, but more modest than the direct genetic effect. So I just wanted to close with a few reflections on why indirect genetic effects matter, because hopefully I've convinced us all that across the lifespan, we find modest but consistent evidence for indirect genetic effects, ranging from nuclear family indirect genetic effects as transmitted via the environment, ranging to peers in adolescence, all the way up to spouses in later on in adulthood. And the reason I think this matters is that the predominant brain disease models of alcohol and drug use disorders largely ignore interpersonal and social factors as it relates to sort of the etiology of sort of where these diseases and disorders come from. And I very strongly believe that if you want to improve public health, investing in relationships and preventive interventions at the family and community level is going to be highly impactful and have pay great dividends. So to sort of borrow a phrase from the epidemiology literature where they say, the society is the patient, I've modified a bit to say that the relationship is the patient. And that we have to think about these etiological factors, tracing all the way back to genomes, but that are transmitted via the social environment when we're thinking about prevention and intervention. One other point that I just want to sort of raise for the group too is that some of the relevant for genetic variation for psychiatric disorders may in fact be localized in partners. And there's some very nice work by Amelie Baud and her colleagues in a preclinical rodent model showing that when you try to disambiguate direct and indirect genetic effects for a series of rodent model amenable addiction-related phenotypes, you find different genes and variants that are implicated. And so, you know, obviously I don't think we're there yet in terms of even tracing all of the direct genetic effects in humans that matter, but broadening our sort of theoretical perspective about how genes impact our behavioral outcomes, I think, is something at least to keep in mind for down the line as we sort of expand our understanding of the of how genes impact the behavioral outcomes we have. And then the last point I want to leave us with is just the importance of the social partner's phenotype as well as their genotype. So I mentioned as we were going through the studies that we controlled for the social partner's phenotype, and we continue to find evidence for these indirect genetic effects. So it's not just that like, oh, your indirect genetic effect is just sort of a weak measure of what you're actually being exposed to phenotypically by your social partner. There appears to be some unique variation. So mapping that to like, what is that phenotype? I think continues to be, you know, a future direction worthy of pursuit. So with that, I thank you all for your attention, and I'm happy to answer any questions. And I'm happy to unshare my slides too to see the folks who are on screen. But if there's also any slides you want me to go back to, I'm happy to do that. So I'll stop there.
Thank you.
Happy to. Yeah, the pout fish. They have quite the series. Yes.
So if genetic effects are indirectly influenced, how are they mediated? I mean, they have to be mediated in it.
Yes, and I think I was both sort of thinking that would be a question that might come up today. And also just in my own sort of musings about like, well, what does it mean? Because obviously you want something that you can intervene upon, right? If you're, you know, evidence that they're there is like only the start, right? And I think this is actually a place that, you know, PHWAS in terms of understanding sort of the environmental architecture of genes that are identified is a place to follow up or at least begin to trace those. But I mean, there are a lot of, you know, what are called hidden regulators embedded in relationships, ranging from things like thermoregulation that like parents provide their young, like that's why they, you know, suggest that when your baby is just born, that it's like skin to skin contact, that's one of the mechanisms of that parent-child relationship for like regulating like biobehaviorally the offspring. Obviously that stops after like a few weeks or months, but you know, I think there's olfactory cues that are embedded in relationships. There's sort of partners contribute to one of those microbiomes. I mean, I think there's really like a whole wealth of avenues that.
There's also the potential to view this in terms of indirect. It's not just risk, indirect. Resilience.
Yeah, and that's so that element, and especially thinking about sort of like gene environment interaction and how even someone with a risky genetic predisposition, if you're partnered with a low risk partner, that actually is quite protective phenotypically. And I think, yeah, adding a genomic perspective to that question would be really interesting.
For someone as naive as me, how big are the genetic direct effects compared to these indirect effects?
They're a fraction. So like when we're looking at the family genetic risk scores, I'd say the effects are roughly half. When we're using a genome-wide polygenic score, I'd say like a third to a quarter. So they're modest. And even, you know, when we're considering all of genetic risk in someone's family tree, you know, it's you know, statistical significance is not always translated to practical significance. So I don't want to oversell the results, but yeah, they're modest.
but intriguing.
Yeah. Oh, yeah.
So, yeah, great talk. What would be your thoughts about ancestry specific traits, given that the sort of stats that
So for the Koga study that I talked about at the beginning with the nuclear families, we looked at this. Koga has, it's a smaller than the European ancestry individual or cohort, but there is a subset of African ancestry families. And so we were able to use, the sum stats that were available at the time to calculate ancestry-specific polygenic scores for the African ancestry families. Now, there was in our model to find indirect genetic effects as mediated through the environment, you have to have an association between, in our case, the family adversities and then the offspring alcohol outcomes. And in the African ancestry cohort, there was actually not, that path was not there. And so that sort of obviated like looking at that indirect pathway. But what I will say is that, I can send the paper / like double check that I'm not recollecting incorrectly, but because we were also tracing indirect pathways through the parental phenotype, because of course, like, one of the things that goes along with in these highly densely affected families with marital disruption, family discord, is of course that many of the families are also have a parent with an active alcohol use disorder. And so we were able to look at indirect genetic transmission through the parental phenotype as well. And if I recall correctly, some of the parental indirect genetic effects in the African ancestry families were significant. And I think that they were significant in ways that for the European ancestry families, they were not. And I think that maternal alcohol problems were sort of an especially potent indirect pathway. But let me also follow up on that. But I think it's a critically important sort of, like both an omission and something that I think that the field is hopefully correcting, but it's definitely has like a long ways to go in terms of getting sample sizes just in general, but also beyond European ancestry cohorts and some stats. Yeah. And, as like many other groups have demonstrated, because the GWAS sum stats are smaller or like in smaller samples, there's just weaker prediction at present.
Yeah. I have a question about the developmental perspective side of this. So, because we know that there are changes in terms of like the magnitude of genetic and environmental effects over time. So I'm wondering if you see something similar with the indirect effects. Like, is there a period of your life where there's kind of the most significant impact of those?
We have not looked at that, but that is a lovely idea. And yeah, I mean, I would guess that it's probably younger. Just based on like, if you're thinking about like, that's the family exposure, and sort of the environment in which that behavior is developing, but no, we haven't dug into that.
In Koga, do you have longitudinal data? Okay.
There is a prospective cohort, or Koga. Koga has like 10 different arms, sort of like I guess 8 different arms, like an octopus. So there is a prospective longitudinal component built into the overall family-based sample. So we definitely could look at that.
Any other questions?
I have a question.
Oh, here, let me stop sharing my screen. Okay, yes.
Hi there. Hi, Jessica. My name is Kira. I'm Rachel's postdoc. Thank you so much for the great talk.
Yeah.
I have a question. So when you're talking about this, and particularly with respect to the parent influences, it makes me think about what types of other support the person who's registered has to fall back on. So I'm wondering, like, did you account for, maybe in the family studies, like, are there other people in the family that the person could turn to, like, extended family or cousins, or have you thought about, like, for the peer studies, how many friends someone has? Like, so if they have, like, one particular troublemaker friend who might be a particularly large influence, do they have other people they can turn to? Or have you thought about like personality types and how that might interplay with the effect from other people? Sorry,
Yes.
another lot of questions.
Yeah, no, I mean, you're thinking along exactly the sort of the track that me and my group are thinking about as well. So thinking about like, well, who's most susceptible to these indirect genetic effects? And in the Swedish studies looking at the adolescent peers, in fact, the individuals with the highest family genetic risk scores are the ones who are most sensitive to being surrounded by peers with these riskier predispositions. And there are much more pronounced effects for drug and alcohol use disorder than major depression and anxiety disorder. In that ad health study that I mentioned at the as part of sort of the background for that, they have a whole part of their study, and I can't remember all the specifics, but they investigate this, and that's a group of sociologists. They always have like fun sort of terms for things. They call it like, you know, investigating like the bad apple effect, and like how like if in a classroom there's just like one bad apple with like, I don't know, a really high polygenic score, like how does that impact like behavioral outcomes in the rest of the class? And now I can't remember the first part of your question.
So I was also asking about is it possible to look at like family size or the number of friends that you have as a predictor of how much other support you have?
Yes. So in the context of the Swedish registries, because there's never actual contact with the individuals in the registries, we don't actually know about you know, we can only make inferences about, you know, like, oh, within type of upper secondary school, you're probably having more interactions with those folks than your peers who like live in the same 10-block radius, or the peers in your same municipality. So we're definitely making inferences that I think would very much merit sort of replication and sort of further investigation of defined peer groups within sort of, you know, measured networks. And Ad Health does have that information. We don't in the Swedish registries. In Koga, you know, we know things about family relationships, and we know from participant self-reports, like, you know, oh, you know, what were your peers like growing up? You know, what was the nature of your relationship with your mom and dad? Was there any other adult that you could count on? You know, if mom or dad were not available. But I would say it's much less refined than someone who has like expertise, or sort of like that's their phenotype, or that's sort of the environment that they like to characterize. So I think there's a lot more to be done. And I think this is a place too that, you know, really demonstrates the need for careful like environmental phenotyping to sort of complement the genotyping and what we can learn from the genetic effects.
Right. Yeah. Really interesting. Thank you.
Yeah. Thank you.